So in this video, we're going to look at the neuron in different segments and talk about what's going on in each of these segments as far as receiving and sending signals. So we're going to start with the receptive segment, and this part of the neuron is both the cell body and the dendrites. And the goal of the receptive segment is exactly what it sounds like, is to receive messages. Now, remember that we have a presynaptic neuron, that's the neuron sending the message, and a postsynaptic neuron. The synapse is that space, right? That part between the two neurons. So the neurotransmitter is that chemical signal that's released by the presynaptic neuron. What we're looking at here is the postsynaptic neuron, right? The one that's receiving those neurotransmitter messages. So when a neurotransmitter is received or binds to this receptive segment, what's going to happen is we have those chemically gated ion channels that will open. Now, later we're going to discuss an action potential. What happens in the receptive segment is different than an action potential. It's called a graded potential. So the difference is a potential is just a charge, right? A charge difference or a change in charge. So the graded potential is going to be smaller than what we will see with an action potential, and it's going to be short-lived. What we'll see as we move on and talk about the next segment, we'll see how all of these graded potentials that are being received, how they get integrated, and then what the result of that is. Now, a couple of terms that we've already talked about a little bit, but I want to make sure we discuss, are depolarization and hyperpolarization. Now remember that for the neuron, the resting membrane potential means that the inside of the neuron is going to be more negative than the outside, and it's about negative 70 millivolts, the inside, or the charge difference. So if a membrane of a neuron becomes depolarized, what that means is there's a change from negative to more positive. So the inside of the cell is becoming more positive. If it becomes more negative, then we call that hyperpolarization. So we're becoming more or hyper negative. Now, it turns out that these neurotransmitters, these chemicals that are received in the receptive segment, these can either be excitatory, so the long term for that would be excitatory postsynaptic potentials, or EPSPs, or they can be inhibitory postsynaptic potentials. Now, what that means is if they're excitatory, they are going to move the inside of the cell more positive, or they're going to depolarize the membrane. If they are inhibitory, they are going to shift the inside more negative or hyperpolarize the membrane. Now, different neurotransmitters, right, are going to be EPSPs versus IPSPs, or excitatory versus inhibitory. Now, there may be multiple neurotransmitters that are binding at the same time, and so the amount of neurotransmitters that are binding is going to determine how many of these chemically gated channels that are going to open on the receptive segment. And then the type of neurotransmitter that's binding is going to determine which types of channels are the channels that are opening going to lead to more negative inside the cell or less negative inside the cell. Are they going to depolarize the cell or hyperpolarize the cell? Now, I want us to think, let me get my pen turned on here. I just want us to think about the different kinds of chemically gated channels that we find in the receptive segment and what would happen if each of those is opened by a neurotransmitter. So let's start with sodium. Okay, if we have chemically, remember the receptive segment, we're talking about chemically gated channels that are opened by neurotransmitters binding, right? So if we have a sodium channel that gets opened, which way is sodium going to move? Does sodium move into the neuron or out of the neuron when that channel is open? So think about, where is sodium more concentrated based on our resting membrane potential? Where do we find sodium concentration higher, inside or outside? Okay, so during resting membrane potential, we find the sodium higher outside the neuron. So that means when those chemically gated channels are open, sodium will move into the neuron. Now, sodium has a positive charge, so if more positives move in, then is that going to lead to depolarization or hyperpolarization? That's going to lead to depolarization, right? Which means it becomes more positive. So this would be an example of an excitatory postsynaptic potential. Now let's move on and let's talk about other ions. So what about potassium? Okay, where's potassium concentration highest, inside or outside the neuron? So potassium is higher, the concentration is higher inside the neuron. So if those channels are opened by the binding of neurotransmitters, then potassium ions will move out. Of the neuron. So if we have positive moving out, then that's going to make the inside more negative, right? So that would mean hyperpolarization. There we go. So that would be an inhibitory postsynaptic potential. Now I have one more chemically gated ion. I'll write it down here. So the chloride ion, what's going to happen, okay, and this may not be something that you've thought about as much, but the chloride ions are more concentrated outside of the neuron than inside. So if those gates are open, the chloride ion is going to move into the neuron. So chloride ions have a negative charge. So if we have negative charges coming in, what is that going to lead to? That's going to lead to more negative inside, or hyperpolarization. Now, in reality, there are multiple neurotransmitters that can be binding. We can have multiple EPSPs and IPSPs occurring at the same time, and when we look at the next segment of the neuron, we're going to find out how those come together and what happens. Okay, now what happens in this initial segment? And so let me get my pen here. And the initial segment is the axon hillock. Okay, so that's that little triangular shaped portion coming out of the cell body that leads to the axon. This is where the summation of all of these different EPSPs and IPSPs will occur. Now, notice that, remember, the EPSPs are leading towards depolarization of the membrane, whereas IPSPs are leading towards hyperpolarization. Now, in reality, multiple signals are being received, and it's at the axon hillock that they're summed together. There's a way that they can be summed both spatially and temporally. And I know this is a little bit small, but if you look at this diagram here below the neuron, this first one on the left, spatial summation, what that's showing you is if you have two different signals coming in at the same time. So for example, this first one, P1, you can see that it is becoming less negative, right? And P2 is also becoming less negative, or moving it towards depolarization. Therefore, the two together then would sum towards even further depolarization. Whereas in this second example, we have two signals that are both leading towards hyperpolarization. So together, they would have a larger impact on hyperpolarization. Now, the final one looks at how one EPSP and one IPSP can basically cancel one another out. So that's receiving signals, right, from two different presynaptic neurons. Now, the temporal summation is when one presynaptic neuron is receiving or is sending signals, multiple signals in a short period of time. So we see here in the first diagram, two EPSPs, right, in rapid succession from the same presynaptic neuron, and those can be added together to lead to a greater likelihood of depolarization of the membrane. And then the two IPSPs then also added together to make it less likely, right, that an action potential would move forward. So in reality, yes, these are both occurring at the same time, and the summation is occurring at the axon hillock. And the determining point, there's a threshold value of negative 55 millivolts. Remember, resting membrane potential is negative 70 millivolts. So that means there has to be a change, right, of 15 millivolts. So a depolarization of 15 millivolts to reach this threshold. So this is an all or nothing thing. If the threshold is met, an action potential will be generated and travel down the axon. If the threshold is not met, then it will not be, the action potential will not be generated. So again, I just want to make sure I make this clear. This is an all or nothing, right? It's not like some action potentials are stronger than others. No, it either occurs or it doesn't based on whether the threshold is met. Now, what is an action potential? And we'll follow along in a minute in detail, but essentially that means that once this threshold is met, it triggers voltage-gated channels to open, and we see the movement of ions, and we'll follow what happens when we look at the next segment of the neuron. So the next segment of our neuron that we're going to talk about is the axon. So, we have our cell body with dendrites. Really bad artwork here, but there's our cell body. And then we have this long axon extension, which we'll see eventually ends in these synaptic bulbs. So it's the axon segment that we're looking at. And remember, when we looked at our summation, what we realized is if the threshold is met, an action potential will be generated. If the threshold is not met, then this would not happen, right? We would not have any voltage-gated ion channels open. But what we're going to follow is what will happen in the axon segment if the threshold is met when you sum, or when the initial segment sums all those IPSPs and EPSPs. So again, the first thing that's going to happen is we have the opening of voltage-gated sodium channels. So remember, when we were talking about the receptive segment, the cell body and the dendrites, those were chemically gated ion channels, right? Those were open because of the binding of neurotransmitters. And the axon, we have voltage-gated channels that are opening, and these are opening because of the threshold of negative 55 millivolts that was met in the initial segment. So we talked about before, sodium ions are positively charged, right? And we have more sodium outside of the neuron than inside. So once those channels are open, sodium then is going to rush in, right? And since sodium is positively charged, that means, that's supposed to be a plus sign right there. When sodium comes in, that means we have a change inside the neuron from negative to more positive. So here's our threshold value, right, that was met in order for an action potential to be generated. Once that happened, the sodium channels open, and we get a rapid change in charge, and that's called, we already know, depolarization, right? When we go from negative to positive. Notice we go to about positive 30 millivolts when those sodium gates open. Now, very quickly, the voltage-gated potassium channels then are going to open. And remember, potassium is more concentrated inside the neuron, so the potassium ions, which are positively charged, are going to flow out. So if we take positive out from the inside of the neuron, that makes the inside more negative. So that process then is repolarization. Okay, so I'm just going to put potassium ions move out. This depolarization, sodium ions move in, right? Now, I also want to point out that an action potential travels along the axon only in one direction. So it starts from the axon hillock and it's going to move only in one direction toward the synaptic bulbs at the very end of the axon. It doesn't reverse directions. And the reason it doesn't is because of what's called a refractory period, which means that segment that just had an action potential can't turn around and have another one. And that occurs because of this moment in time called hyperpolarization, where the potassium channels that have opened cause this to be more negative than negative 70 briefly. And there are two parts of the refractory period. The first is the absolute refractory period that occurs first. At this point, absolute meaning another action potential is impossible. It cannot happen. Immediately after that is what's called the relative refractory period, which means it is harder than normal to generate an action potential because the membrane is still hyperpolarized, but it is possible at that point. Again, the result of this is so that once an action potential is generated, it generates the action potential in the neighboring section, in the neighboring section, in the neighboring section, and it travels down the axon, but only in one direction, and we will not get it moving both directions back and forth. So it's important when we're talking about the action potential to understand which ion channel is opening to cause depolarization, which is the sodium-gated and the voltage-gated sodium ion channels. And then immediately the voltage-gated potassium ion channels open, and that causes the repolarization and ultimately the hyperpolarization of the membrane. One more thing I wanted to mention, we talked about which ion channels cause depolarization and repolarization. After hyperpolarization, so the potassium ion channel is what leads to hyperpolarization. But then the correction back, right, to the negative 70 millivolts, that is caused because of the sodium and potassium leak channels that will bring that back to the resting membrane potential. Okay, so as the action potential moves along the axon, there are two different ways that it travels, and this is based on whether or not the axon is myelinated or not. Now, just as a review, we have a little bit different process of myelination in the peripheral nervous system versus the central nervous system. Can you recall what is the type of glial cell that's responsible for myelination in the peripheral nervous system? So it's the Schwann cell, right? And the Schwann cells, remember, they wrap that sort of their fatty, lipid-rich plasma membrane around the axon. Now, what about in the central nervous system? What is the special glial cell that's responsible for myelination in the central nervous system? So those are the oligodendrites. Now, let's look at, so if an axon is myelinated, remember that there are spaces in between the myelin that are called the nodes of Ranvier. And so in this figure, right, we see, a node here and here and here. And what happens, so we call the movement of the action potential when along a myelinated axon as saltatory conduction. And essentially what you can think of is this myelin serves as like an insulation. So the voltage-gated sodium and potassium channels only have to open in between the myelin. So only at the nodes does this exchange of ions have to occur. So essentially the signal then you can think of as kind of skipping node to node because that's where those voltage-gated channels are opening. So because of that, it's a much quicker movement along the axon. Now, if an axon is not myelinated or unmyelinated, it has to continuously travel along the axon. So, in other words, at each position along this axon, all the sodium and then potassium voltage-gated channels have to open. Therefore, we have slower movement of that action potential. So the last segment that we'll talk about with the neuron is the transmissive segment. And what occurs here, the function of the transmissive segment, is the release of these chemical messengers or neurotransmitters. Now remember, the very end of the axon we call the synaptic knob, or we could call it the axon terminal. And what we see here is we have vesicles that are present in these terminals, and the vesicles contain our neurotransmitters. Now, what's happening before the action potential reaches the axon terminal is that calcium ions are pumped out of the synaptic knob into the interstitial fluid. So in other words, we have more calcium ions in the interstitial fluid, right, than inside the synaptic knob of the neurons. As that action potential travels down the axon and it's received, right, at the axon terminal or the synaptic knob, what happens is it depolarizes just like the rest, right? You have voltage-gated changes because of the change in charge. Now, when it depolarizes, it is voltage-gated calcium channels that are going to open. Now, since calcium was more concentrated outside, then that means calcium is going to flow into the synaptic knob. Now, what will happen is those calcium ions actually bind, and they cause the synaptic vesicles to travel to the plasma membrane, where they perform a type of excitatosis, where those neurotransmitters are released into the synaptic cleft. Now, I just want to review something for you. So remember, the synapse is the point between, right, the presynaptic neuron and the postsynaptic neuron, or an effector. That's the synapse. The synaptic cleft is this space. Remember, they're not touching, right? So the space between the sending neuron. So now what we're looking at is the presynaptic neuron, right? The one that is releasing the neurotransmitters. The postsynaptic neuron is the one that is going to bind those neurotransmitters. So the synaptic cleft is between those two. So these neurotransmitters then are released in response to the action potential reaching the synaptic knob or the axon terminal. And what happens when that membrane is depolarized, those calcium channels, the voltage-gated calcium channels open, calcium flows in, that causes the release of the neurotransmitter into the synaptic cleft. So we'll talk a little bit about types of neurotransmitters, some of their actions in this lesson. We'll continue to learn more about them as we continue our discussion of the nervous system. So we can classify neurotransmitters based on their chemical structure, what their structure looks like, and then we can also classify them based on their function. So we have over here on the left side a classification based on their chemical structure. So we're going to look at acetylcholine in the next slide, specifically its action. So we'll talk about it more and more as we go through the nervous system. So that sort of has a category of its own. Then we have what we call biogenic amines, or these can sometimes be called monoamines. So just because I want you to start thinking about these and making connections, I'm just going to write here monoamines. So that's just another word for biogenic amines. And these we can classify as catecholamines. So let me see if I can write this here. Whoops. Okay. And then over here on this other side, we can say indolamines. And those probably don't sound familiar to you. Whoops, I don't know what happened to my... That's supposed to be a CH right there. So catecholamines may not sound familiar to you. However, probably these things that are types of catecholamines do sound somewhat familiar. So dopamine, epinephrine, norepinephrine, so when we talk about the autonomic nervous system, we'll talk about those more. And then again, indolamines, that term may not sound very familiar to you, but probably histamine is something you're familiar with, and serotonin. Now, one of the first drugs, medications for depression, or one of the first classes of drugs to treat depression, were called MAOIs. So MAOI stands for monoamine oxidase inhibitor. And so the way neurotransmitters work, they are not around a long time, right? They don't hang around in that synaptic cleft a long time. They either get what we call reabsorbed, or the reuptake occurs, they get taken back in by the sending neuron, or they get broken down by an enzyme. So that way they have an action, right, for a period of time, and then they don't until the next action potential is sent. Well, the MAOI, the monoamine oxidase inhibitor, it inhibits the enzyme that breaks down or that metabolizes these monoamines. So in other words, the effect of that medication would be to allow those specific neurotransmitters to hang around in the synaptic cleft for a period of time. Now, the other chemical structure, amino acids, some of them can serve as neurotransmitters, and then neuropeptides, so small peptide molecules. Now let's look over here at the classification of their functions. Okay, so we looked at structure. Now let's look at function. So we can look at the effect they have. So some can be excitatory, right, or leading a graded potential towards more positive. Some can be inhibitory, so that would be leading the graded potential to be more negative. Now, the action, so neurotransmitters, when they bind, they can bind and directly open an ion channel. That would be direct action. Or by binding, it can be indirect because it sort of has a second messenger. So they bind, there's a little bit of a process before the ion channel is open. So again, we'll talk more about neurotransmitters as we go through the nervous system, but I want to specifically in the next slide look at acetylcholine. So let's look at an example of a neurotransmitter. This is acetylcholine, and so we might as well start learning that we will abbreviate acetylcholine regularly with just capital A, capital C, H. So we'll use that to abbreviate acetylcholine. So this is our presynaptic neuron, right? And the action potential has traveled down the axon. It's reached the synaptic knob or the axon terminal. And notice that in the vesicles, the acetylcholine neurotransmitter is already present in those vesicles. Once the signal is released, does anybody remember what channels, as this membrane gets depolarized, what channels will open? So it's the calcium ion voltage gated channels open. Calcium then flows into the synaptic knob. That triggers the release of these vesicles to release the acetylcholine neurotransmitter into this space, right? This space has a special name. It's called the synaptic cleft. And once it's in the synaptic cleft, there will be receptors in the membrane of the receiving cell. So we haven't talked about the muscular system just yet, but one of the cells that actually responds to, has receptors for acetylcholine, are skeletal muscle cells. So we call that a neuromuscular junction between the neuron and the receiving muscle cell. Acetylcholine is also present in our autonomic nervous system. We'll learn more about that when we cover the autonomic nervous system. But there are more than one type of receptor that acetylcholine will bind to. So one of those is called a nicotinic receptor, and the other here that's shown is a muscarinic receptor. Notice that the nicotinic, in this case, it's a direct. So once the neurotransmitter acetylcholine binds, it opens the ion channel directly. However, the muscarinic receptor, when it binds, there's this second messenger, this G protein, that has to actually cause something to occur. So it can be opening an ion channel, it can be signaling another molecule, but it's not a direct action. It goes through a second messenger. Now, another thing that I want us to see is that the acetylcholine is released into the synaptic cleft, but it shouldn't stay there indefinitely, right? There needs to be a way to remove that so that it's not always triggering a response by that receiving neuron or muscle cell. So one other important thing I wanted to point out is this enzyme right here, acetylcholinesterase. So this enzyme is responsible for, once the acetylcholine is released into the synaptic cleft, this enzyme then metabolizes it or breaks it down into choline plus acetate so that we don't have this neurotransmitter indefinitely present in the synaptic cleft. That would not be a good thing. Now, as I mentioned previously, neurotransmitters shouldn't hang around, right? They should only be around for a short period of time because we want that to be in response to a stimulus, right, that's occurring. So we want to control when we move our muscles, right? We don't want that to constantly be causing us to have muscular movements that we don't want. So the neurotransmitters can be removed in different ways. So this enzyme is an example of one way, right, that they can be metabolized or broken down in the synaptic cleft. And I mentioned to you earlier the MAOIs, those medications actually target an enzyme that would break down some of those monoamine neurotransmitters at the synaptic cleft. We also have the reuptake of neurotransmitters back into the sending or the presynaptic neuron. So that's one way that those are removed. There's another type of medication that also treats depression called SSRIs. So that stands for selective serotonin reuptake inhibitors. So in other words, those medications are slowing down the process of the serotonin being taken back in by the presynaptic neuron. So the effect is to allow the serotonin to remain in the synaptic cleft for a longer period of time.